Bismillahirrahmanirrahim. I am Dr. Tahir Mahmood Qureshi, serving as an assistant professor at the Department of Food Sciences. I did PhD from Norwegian University of Life Sciences, Norway. The title of my presentation is Characterization of Value-Added Dairy Products. Here is a layout of my presentation. First of all, I would like to explain value addition, then characterization, then physiological functions of milk-derived bioactive peptides, then characterization of some value-added dairy products, and finally conclusion. How value addition is done? The first point is to make unadulterated dairy products. This is also called value addition. Adding some specialized ingredients, especially fruits, pulps, spices, herbs, and their extracts, addition of probiotics. A removal of fat from milk is also called value addition, then standardization. Standardization means adjustment of fat levels into the milk so as to make different kind of dairy products afterwards. Employing processing methods, pasteurization, ultrasonication, etc. We can also call these as value addition. And why value addition is done? The first point is to enhance organoleptic properties. In organoleptic properties, it means we can enhance color, appearance, texture, flavor, and some other appealing characteristics, making functional foods with more health benefits, nutritive value, to extend the shelf life of the product, and in this way we would have greater economic gain for industry or dairy farmers. Adding value to raw milk. What does it mean? It means we can produce pasteurized flavored milk from raw milk. We can produce yogurt, dahi, cheese, paneer, ice cream, frozen desserts, and sweets. Yogurt and dahi, they are two different things, but we can also say that these would be more or less similar, but in composition wise, when we talk about the manufacturing of these products, they would be different. In dahi, we do back sloping technique. It means we have traditional technique of making dahi by taking already prepared dahi into the milk, and then we can set that milk at 40 degrees centigrade or more or less 42 degrees centigrade in order to make dahi. But in yogurt, we use pure cultures. It means if we have Streptococcus thermophilus and Lactobacillus delbrueckii subspecies bulgaricus, it means we are adding pure cultures, and in this way we can prepare yogurt, and these cultures are being produced and they are used at industry. The next is the cheese and paneer. Paneer is actually acid and heat coagulated product. We can use citric acid, lactic acid, citrus fruit juices for the preparation of paneer. We add into the milk such kind of acids, and then coagulation occurs. Then after filtration by muslin cloth, we can prepare this paneer matrix. But in cheese, we use pure cultures and rennet enzyme for the coagulation of milk. In this way we can prepare cheese finally after pressing. Ice cream may also be called frozen dessert, and we can also produce different kind of sweets in our culture by using raw milk. Characterization of products. Under physicochemical test, we can say that we can determine moisture, dry matter contents, we can determine pH, acidity, protein, fat, ash, and we can also do sensory evaluation. Under antioxidant potential, we can use different kind of assays concerning total phenolic contents, total flavonoid contents, radical scavenging activity by using DPPH and ABTS radicals, and then ferric reducing antioxidant power. We can also characterize the products concerning AC inhibitory activity, which is antihypertensive effect. The detail of these antioxidant potential and AC inhibitory activity is coming ahead in some other slides. Physiological functionalities of milk-derived bioactive peptides. Actually, milk contains two types of protein. The first is casein and the second is whey. Casein comprises of alpha, beta, gamma and kappa casein, while whey protein comprises of alpha-lactalbumin, beta-lactoglobulin, immunoglobulin, serum albumin, lactoferrin. During ripening, fermentation and digestion of dairy products or milk, bioactive peptides are released. These bioactive peptides have different kind of functionalities concerning antioxidant activity, antihypertensive activity, immunomodulatory activity, antimicrobial activity, nervous system, metabolic health and bone health. But in this presentation we are interested in antioxidant potential and antihypertensive activity. Free radicals are reactive oxygen species. These are produced in our body by different kind of factors, like for instance stress, irradiations, toxic substances, ingestion, body's metabolism and inflammation. And we can also expect more and more free radicals due to oxidation of fats and oils. It means in rancid fats and oils, we may expect more and more free radicals or reactive oxygen species. And we can also expect such kind of free radicals or reactive oxygen species in those products which have been fried over and over again. I mean locally or culturally, if we are consuming those products, like for example fish, samosas, pakoras, it means in our culture we are getting these from the shops, and the oils or fats which are being used for the frying of these products, they are being used, those oils, they are being used over and over again. In this way, more and more free radicals would be expected in those products, and we may also take more and more free radicals from those products. If we have these free radicals in our body, if we take these free radicals from the products, then these free radicals may cause cellular tissue damage, and these conditions may lead to cancer, aging, heart disease and neuro disorders. But if we have antioxidants naturally present in our food products, then we may expect that these free radicals would be scavenged and we would be saved from such kind of diseases. Actually, the chemistry behind these free radicals, when they form, they lack electrons. But if we are taking antioxidants in our diet, these antioxidants, they donate those electrons which have been missing in free radicals. In this way, these free radicals would become more stable, and they would not give any kind of chain reactions in our body, and we would be safe. We may also use synthetic antioxidants in oils or fats just to stop oxidation of those fats and oils. In this way, we would also expect that there would be no rancidity, and there would be no smell in our fats and oils, and we would also expect less free radicals in those products during aging process or during storage. Here is one of our study regarding physicochemical characteristics of colostrum. In our culture, Indo-Pakistan, this colostrum is locally known as boli or khis. We observed that in the colostrum we have solid not fat, protein, ash and fat at very high concentration in the start of the milking. But we have observed that during subsequent milking, these contents would be reduced, and these contents would be normalized after 5 or 6 days, as we can observe in the buffalo milk. And pH was also very low in the start of the milking after parturition in the colostrum. Then this pH would be increased and it would become normal after 6 days or 7 days. Concerning antioxidant potential, we have observed that the antioxidant activity was very high, the values were very high in the start, but these values would be decreased after subsequent milking of the colostrum. You can see these values of total phenolics, reducing power, DPPH, radical scavenging activity, and Trolox equivalent antioxidant activity. The values of these were very high in the start. And this antioxidant potential was owing to the presence of superoxide dismutase, glutathione peroxidase, catalase, lactoperoxidase, lactoferrin peptides, and other casein hydrolysates. We can expect antioxidant potential due to presence of these compounds, which are naturally present in the colostrum as well as milk. Here is another study concerning paneer incorporated with spices. It has been reported that bioactive compounds are present in the spices, and the main intention here, or the main emphasis, or the main focus to carry out such kind of study was to increase the bioactivity of paneer. And another motive behind such kind of paneer is that we can also expect organoleptic properties which would be increased by the addition of spices. It means for our culture, if we consider that paneer is a tasteless entity, then by the incorporation of spices we can enhance the taste of that paneer. You can see total phenolic, total flavonoids, and reducing power, they were very high in the spices. Here is another publication, paneer with date fruit extract. The main emphasis was here to increase the bioactivity of paneer as well as whey by the incorporation of different date fruit extracts. Next is the ACE inhibition and peptides. This is one of the study which was carried out at Norway regarding angiotensin 1 converting enzyme inhibitory activity of the Norwegian autochthonous cheese Gamalost and Norvegia after in vitro human gastrointestinal digestion. Here is renin-angiotensin system. Liver produces angiotensinogen, which is converted into angiotensin 1 by the help of renin, which is produced from the kidney. This angiotensin 1 is converted into angiotensin 2 with the help of angiotensin converting enzyme, which is secreted from lungs. And this angiotensin 2, it causes vasoconstriction, and in this way blood pressure rises. But if we inhibit ACE, then there would be no conversion of angiotensin 1 into angiotensin 2. Then there would be no rise in blood pressure. It means blood pressure would remain stable. And we observed high ACE inhibition in our cheeses, and we also observed ACE inhibitory peptides in our samples. We also carried out some other studies regarding in vitro digestion and processing of cheddar cheese, and we observed that if we have in vitro digestion, bioactivities of those cheeses would be increased, and if we process the milk by sonication, microwaves, and high pressure processing, then we observed that bioactivities would also be increased. Here is one study regarding shelf stability of rasmalai. We observed that shelf stability of rasmalai was increased by the addition of different preservatives in permissible limits, and we also observed antioxidant potential of mango pulp and we observed that by the application of ultrasound, antioxidant potential of mango pulp was increased. In conclusion, we can say value-added dairy products would be healthy and delicious, and the infrastructure which we have at our department of food sciences, we can explore antioxidant potential by using different kind of assays, like for example total phenolic contents, total flavonoid contents, radical scavenging activity, and ferric reducing antioxidant power. Thank you so much for your kind attention.